I'm excited to be here today to share some of the work we are doing in the Mara and also some of the projects we are currently working in Serengeti Mara ecosystem. So the topic of today is the spatial and temporal shift, a decade of macroinvertebrate assemblages in tropical river and the implication in well monitoring. Most of you, I'm sure you work in terrestrial ecosystem, but I want to give you different lens of also working in freshwater ecosystem where we are all working towards the same goal of conserving ecosystem biodiversity and things like that, and how the terrestrial and aquatic cuts across. This is who I am. I identify more as a taxonomist. I do a lot of identification for macroinvertebrates for the fish. I identify myself more of ecologist. I study the trends of diversity and things like that, and also interested with the aspect of trace element and these emerging pollutants in water. Before I talk today's topic, which is one of my main chapters for my dissertation, I'm going to just quickly show you what else we are doing in the Serengeti Mara ecosystem, mostly in the Mara River, and what kind of projects we are working there. As Subalaski Lab, Amanda Subalaski, she's my advisor from the University of Florida, and we have a research camp in Masai Mara around Mara Serena in Kenya. And there we are currently focusing on some of the projects where we want to quantify the concentration of trace element in Mara River, because this river flows from Kenyan side all the way to Tanzanian side. And there's a whole aspect when the river enters Tanzania, there's a whole place around Tarime where is large scale gold mine. And as we all know that they use mercury as an element for amalgamation. So the photo on the left, extreme left on the top, we took that photo during last year, during summer when we were sampling. So those are some of the massive pipes are coming from the artisanal, from the industrial gold mine. They say they treat the water, but probably the results might show differently. But so this is somewhere upstream of these activities happening. All these is happening a few meters. This photo and this were taken the same day. So we have these pipes happening downstream. We have people fetching water, using the same water to clean utensils. There's active fishermen. They are also catching fish. And others. Those are some of the fishes we are getting in the Mara. And what that tells you is that because all these fishes are big fish and people are fishing in the Mara for economical purposes, if the fish is able to bioaccumulate this trace element in their bodies, such as mercury, for instance, over time, because this fish is almost 15 kilograms, which means it has been in the system for a long time, right? And if people end up catching that fish and they eat it, you are able to also acquire those contaminants in your body as well. So this is another chapter of my dissertation about trace element in the Mara. We have an active project where we are tracking this particular species of fish. It's called, the common name is Ningu fish. Ningu should be a Maasai name, if I'm not wrong, but the scientific name is Labeo victorianus. 2 years ago, the fish was listed under IUCN, threatened, critically threatened endangered species, but now the status has been reviewed because the ecosystem or the watershed catchment has improved over time in the Mau, and probably is not a threat. But we're interested to see this fish from the literature is said to be living in Lake Victoria, and it swims upstream all the way to Kenyan side for spawning and a lot of activity. So we want to see how far could this fish swim from the lake all the way to the Kenyan side. And we usually insert some trackers on the fish. We have those tracking system on the land. They are able to communicate with the tracker, and when the fish either swims upstream or downstream, our gadget here is able to download the data of what tag number that is either swimming upstream or downstream. And that's how we are able to analyze and say, oh, this fish can swim this X kilometers and things like that. We're also doing a lot of water monitoring programs in the Mara. We have stations there in the Mara which periodically they give us information using this gadget called the Manta probe. It gives us information on water quality, things like dissolved oxygen, temperature, turbidity. Just like our friends are doing with the Earth Ranger, we are able to get live details of what is happening in the river in terms of these parameters in water, or if the discharge changes and things like that. Because as I'll be speaking, the Mara River is really important. Without it, probably we won't have the Serengeti Mara ecosystem and things like that. We're also interested with aspect of microbiome. These are the study of bacteria, especially the gut bacteria in both fish, hippopotamus, because their presence or absence might modulate how an animal might behave or how fish really behave and things like that. And just studying different aspects in terms of isotopes in these ecosystems, things to do with DNA and also the fish gut, kind of some of the analysis. That is all what we do in the Mara. But today we're going to be talking about this particular topic, which is, I've said, my first chapter of my dissertation. And the reason why is because we really have limited studies on the spatial temporal kind of comparison. A lot of people just do a study and that's it. But we are able in this study demonstrate that whatever was done in 15 years ago, 2002 on 208, 209, that's our historical kind of data. And we have the present data from 2021, 2022. So we wanted to show what has changed over time in this catchment in terms of land cover, in terms of water quality, in terms of biodiversity in this catchment, both of these insects. So this time span is rarely studied by a lot of people because we don't have people who really keep the data so well. But I was fortunate because my advisor worked in this catchment 15 years ago, and now I happen to be studying the same catchment, and we have almost the same data. So I'll be comparing that kind of data and see how we can use it for this terminology called biomonitoring, which is an aspect of using these insects in water in order to infer or tell the water quality in it, all right? And so I'm aware you all know this concept as people studying the terrestrial ecosystem. A lot of studies are now trying to bend this curve, trying to avoid animals from going to extinction. I think one of the RISE core objective is trying to let animals not go to extinction. But we have a challenge with what I study because, whatever I study is less studied, and under IUCN it's DD, the data deficient kind of taxa. So for a lot of giraffes, rhinos, the status is already known. But what I study, it's less studied. People don't want to do what I'm doing. And it's difficult to even bend the curve because you don't even know what we are, right? And then comparing the terrestrial ecosystem and freshwater is that we have already a lot of biodiversity decline is somehow expected to, it's more, species are more into decline, even in this freshwater ecosystem compared to their counterpart, which are terrestrial. Because the freshwater ecosystem, there are a lot of factors which are coupled and working all together to drive species to extinction as compared to the terrestrial ecosystem. And some of those factors include those discussed there. And this threat continues a lot because globally, as you all know, we're dealing with the aspect of climate change. We're all worried about 50 years to come from now. And if this freshwater ecosystem, then they are understudied. And again, the species in those ecosystems are even more threatened. How about when we also couple it with climate change? That means then this ecosystem will even decline further, even losing more species in the wake of climate change, because we are currently in the tropics. And as we all know, along the equator, we might experience more heat compared to those countries far north or far south. Someone might be asking themselves, why the Mara River? Why not any other river system in Kenya or in Tanzania, or I don't know. The reason why the Mara River, this is one of the major transboundary river between Kenya and Tanzania. It's a river of international importance, drains to Lake Victoria. Lake Victoria is the main source of water all the way to Nile River. So really, it's a river of importance. This is the only river where annually we have this great migration of the wildebeest happening. We have the mega faunas, like hippos and things like that, and also really charismatic species we can find in this river. And so continuing this story is also like, take for example the Serengeti Mara ecosystem and compare the two. You'll find out that even a lot of tourists who come in this catchment, they are always going mostly into the terrestrial animals. They want to see big charismatic rhinos, elephant, leopard. Everybody is on this extreme left. But how about this river, which cuts across this ecosystem, and it's really important? How are people really doing anything about this river not, drying up? And because it's really important river system for this catchment, which drives tourism. And in Kenya, I think tourism brings around 30% of GDP in the country. So without this river, really will have messed up and probably even gone low in terms of GDP and things like that. I'm also for sure thinking Tanzania also depends a lot on tourism in Serengeti and all that. So it's really a river where we need all combined effort and conserve this modern system. I'm going to introduce to this aspect called the river continuum concept for those who are new in aquatic system. What you are seeing on the left, this flow chart tells you that as you move from the headwaters of the river all the way to the mouth of the river, assuming this is the Mara River, this is the mouth forest, the productivity inside the forest is low. The light cannot penetrate through because the canopy is really covered, right? So what means the main source of energy for this river in the more contained areas will be materials coming from outside into the river, which are referred to as alloctogenous material. This might be leaf, the leaf falling into the river, which forms the basis of food for this river upstream. But as the river flows downstream from the forest, somewhere in Kenya, then the channel becomes wide, less trees, the light can penetrate to the river. The river is able to do its own primary productivity, produce algae and things like that. They're going to be colonized by the small insects we are calling macroinvertebrates. Then the fish comes in. So the chain continues. So the only difference is that at the source of the river, the canopy is really so dense, trying to prevent the light from getting in. So the main source of two energies of a river, alloctogenous coming from outside the river and autochthonous, which means the river itself being able to produce the energy it needs, light coming in, algae and all that kind of stuff. So as we move by, I'm testing this kind of concept in the Mara River where this catchment has issues of land use. Already we have issues of wildlife and livestock subsidy where every year as wildebeest crosses the Mara River, we have thousands of them which drown in this river catchment. They are decomposing within the river catchment. What does that then do to the water quality? What does that then do to the biodiversity of this ecosystem in the river? And also, every river has this aspect where there are people just cutting down trees or there's a lot of pollution coming from external sources. How are these three factors then really impacting what I'm studying? And so we wrote this book chapter in this new book called Afrotropical Streams and Rivers. This came out last December. It's an active book. This is where you can get it. But it addresses all the stressors which are within this river catchment. I'm studying the Mara River. What is really driving the Mara River negatively? What are the issues we are discussing there? So this is one of the book chapter which contains a lot of that. But I'm going to be splitting all what we discuss in this chapter in the few slides. And so we are really interested with these three major issues where we have land use change as an issue in the Mau, where the trees were cut down 10 years ago compared to now. Issues of wildlife subsidies increasing in the river, depending if the animals, the wildebeest die while crossing or not, or issues of hippopotamus, which bring in subsidy into the river, right? And also issues of point sources of pollution, and how those three really affect our organisms there. And some of the evidence from the catchment is that land use is a problem for sure. There's a lot of documentation about what is happening in this catchment. This is somewhere where inside the forest where this river comes, you can see even there's an active agriculture activity happening somewhere inside the forest. And that has really impacted the river catchment in a way that the water of the Mara River throughout the year is this color. That's an indicator that a good river really should be clean, right? There are places you go and you appreciate how clean rivers are. You look into the river, you are even able to see even fish swimming. This is not the case with our river here. And it has been like this for the past probably 10 years or even more, stretching back from there. I'm sure 100 years ago, 50 years, the river wasn't like that. But because of what is happening in the catchment, this is the trend throughout the year. The water is murky, turbid, and all that kind of activities happening. There's a lot of evidence about point sources of pollution, how mercury is getting to the Mara River. There's evidence about wildlife subsidies in this river catchment and what that does. As you can see here, these are some of the wildebeest which were saved by the river after drowning. And now they are decomposing within the river system. You can see vultures coming in, eating them, tearing the flesh. But at the end of the day, there are going to be a lot of probably nutrient bleaching into the water. And the bones themselves from the wildebeest take a long time to break down that nutrients, and bleaching can take longer than expected. And so I'm going to introduce you to our study area. This is the Mara River watershed. These are the sites I've been studying in the Kenyan side. This is our transboundary border, and this is Tanzania. By this time, I'm going to be presenting data from 2021, 2022, 2023. We are still applying for permits in Tanzania to get access to this part, the Rukoste, Tawiri, and the rest. We got the permit last year. So these analyses do not include data from the Tanzanian side because we haven't yet analyzed those. And this is what it looks like in terms of land cover in this area, where one site where the river, what we are calling our site one, is really somewhere deep in the forest. And the rest of all the sites though are outside the forest. So you can see the interpretation of all this, what this map looks like. As you can see, there's a lot of also cropland happening, even further upstream. There are people doing agriculture somewhere in Kenya up there. And that's why the river can never have clear color, because if there's agriculture going somewhere there and it's somewhat deeper in the forest, that means somewhere deep in the forest, or not even in the forest, it's outside the forest, but it's all the same catchment. So if people are farming somewhere here and it's raining, everything is being washed to this drainage basin and the water can never be clean. This is the map of what the rainfall in this region looks like. With higher, this is a mean or an average for past 20 years. And you can see this area, which is our upstream sites, really receives heavy rainfall compared to the rest of the sites as well. And so, told you I will be comparing data 2 years each in different time span. This was collected during this time period by my advisor, that's Amanda there on the extreme left, and Chris, Amanda and Chris, and I'll be also doing the same. So we employed the same technique. We tried to reproduce the study, and we did the same thing during these years. I've done the same thing. And those insects, which I'm calling macroinvertebrates, are collected by this net, which is called a kick sampler. And those are some of the insects you can see. They are called macro because you can see them with your naked eye, but sometimes are really tiny. You need the aid of microscope to be able to see the. And this is how they look like. Probably you have never seen anything like this, but in fresh water, in rivers, just the way you have elephant, rhinos, and everything else, we have all these different biodiversity under the water, which you'll never see and experience. Probably in, I don't know, when you go fetching water, it's difficult to find them because they are launched in stones, they are launched on the soil sediment. So you have to disturb the river in order to see them, right? So how many had not seen this until today? I think a lot of you had not seen this, right? So we have a whole biodiversity ecosystem happening in rivers. Pay attention to the first, is it column or whatever it is, but it's all this. So these have an acronym which is called EPT. EPT is an acronym for mayfly, common name, stoneflies. So that's a mayfly. This is a stonefly, and this is the caddisfly. So these three have been used for a long time all over the world to monitor the river systems everywhere. In South Africa, they have the SAS. In European system, they have a different, I can't recall the name. In the US, they also use the same technique, but with a different name. In Tanzania, you have TARIS. Probably you don't know about it, but it's written by Dr. Lulu Kahaya. She's now at the University of Dodoma. TARIS is an acronym for Tanzania River Scoring System. So she has intensively sampled rivers in Tanzania, and they have been able to document the taxons of these organisms here in Tanzania. In Kenya, we are trying to do the same by a project called KISS, being led by Dr. Masese from University of Eldoret. But yeah, these are some of the ones. But the ones you see on the lower side, when you find them, then it indicates that, well, the water is not good. They love to live in all the extremes. They can be in fresh, they can be where the water is clean, but mostly when you find them, they are where the water is not so suitable. For instance, in a river where we have watering point for wildlife, and they come, they poop there, and they go. You'll find a lot of these taxa in there. They are more opportunistic. They want to encroach that kind of an area where it's more nutrients and things like that. And so I'll be introducing you to some of the results so far we have. And my bad, but for the land cover classification, we don't yet have the data. We are working on it, but what we have done so far, different from how people calculate land cover in rivers, is that we are calculating every point as an area drainage. How they have done it traditionally before is that when you have a river flowing, people usually calculate land cover at a buffer of some meters from the river, like 5 meters. 5 meters. The problem with that is that you are going to get the influence of a different land cover, which is not in your own drainage catchment. I don't know if you're getting what I mean. For instance, if you take M4, it will tell us this site, for instance, this site, the drainage basin is all this, M1, all of M2, all of M3, and then all of this M4. In order to get the drainage of this basin here at this site, we have to consider this catchment, that catchment, this and that. That is the only different thing we have done. Instead of coming to this site and calculating 5 kilometers across this river, if we did that, then it will only give us catchment of this area, and we will miss which is the main drainage area from the source. You get that? Yeah. So we are currently doing that. The results will be in 6 clusters, which are like forests, cropland, and things like that. But this paper is actually due in 2 days' time, which is 31st at night. And we are working on it. It will be going to freshwater biology. But I have trust with my team from University of Groningen, and they're able to give us the results before then. For the environmental variables, which is mostly stuff to do with what is inside the water, what is the level of nitrate, what is the level of ammonia, what is the level of all these different. We compare these two, and whatever will be popping out from the right side is what was different in this within 15 years. This is what changed. So in 15 years' time, we are able to see that ammonia somehow, earlier 15 years ago, it was higher, but now it's a little bit lower. Conductivity, which is measure of how water is able to conduct electricity or something like that, has increased over time if you compare with this graph. So all these are proxies of what is happening in the upper catchment in terms of land use change, because conductivity, stability, nitrates increasing, dissolved organic carbon, that's the acronym, and TDC is total dissolved solids. They have increased within a period of 15 years, which means a lot is still happening in this major catchment, which influences the quality of water in this area, all right? So before I show you the results of the diversity, it's good I show you what is happening in the catchment in terms of my stressors. That is one, land use, which I showed I'm interested in, and also anthropogenic activities, which affects water quality. And we want to see what are the results of this water in this 2 time period before you even go to the communities themselves. Going to how the communities changed over time. Whatever happened 15 years ago, I've called it historical. What is happening now, because I'm sampling now, it has the time period that there shouldn't be a problem. So interestingly, how you interpret this plot is that The cluster in 15 years ago, all the sites were less altogether. And now the sites have diverged and moved away, which means these sites were almost the same because they had the same composition, right? They had the same species richness. But over time, in 15 years ago, these sites have diverged from each other, which really is a proof that this has been a lot. And we tested this with postal analysis, and we have seen that this distance here, which we are measuring, is actually significant. Yeah, things have changed for sure. And even how far they have diverged from each other is also significant. So this is telling us the composition of the species from 15 years ago as compared to now, yeah, for sure. Things have changed, but we don't yet know, did it change for the better or for the good? Because it is probably not how it used to be. That's the next thing we need to ask ourselves. We also wanted to see in terms of diversity of this composition of these insects. Same color coding. Historically, these are the plots down here, which indicate that 15 years ago, the diversity was probably lower as what we are finding now. So in freshwater ecosystem, there's something tricky. An increase in species in freshwater probably would not show how good an ecosystem is. You know what I mean? Because some of these species are opportunistic. They will come in there if you have poor water, exactly. If you have pollution, then some of the species which used not to be there 15 years ago, the ones I showed you will colonize some of the sites in the river. And I think that is what is happening here. So this increase in diversity doesn't in any way infer good ecosystem in water. So it depends what increase. But yeah, I wanted you to get that point. We went further and wanted to study this composition of the communities. This is the historical data, and this is the present data. This is still a support of the clusters were all together, which means the bigger cluster, all this, in all the sites we sampled, they almost had the same composition. In this current site we are sampling, this composition, the river upstream is almost completely different from the river downstream. Does that make sense? Mm-hmm. This dot, blue dot, is almost different from the red dot. They are all like distinct. So which means if you are interested to learn these insects, then you have to sample the whole river, because upstream, the communities are completely different from somewhere downstream. And that's what we are calling. How then, if these things changes, how do they impact the usability of these insects for biomonitoring? All right. Those are some of the things that we speak. So we are also wanted to see these communities of insects. The community thing, I'm using the community term because in insects, we don't identify them until species is complex. So we just leave it at taxa level, which might be a family level or a genus level. And a community means all whatever we form, the species richness. That's what I mean by my community terminology here. We wanted to see, because things have changed over time, this and that. What drove this big change in the present composition? Does it make sense? We have seen in the current period, the sites are almost away from each other. What is driving that change? So we conducted this analysis here to understand what is really driving the macroinvertebrate composition in the present time. We measured 14 different variables and used this model, that canonical correspondence analysis. And at the end of this model, these only five, 1, 2, 3, 4, 5, only these 5 variables out of 14 were the ones which were significant with the composition of the macroinvertebrates in every site. And which are those? Dissolved oxygen, elevation, as you know, altitude, as we go upstream where the water comes from, which is almost 28 meters above sea level, the composition will be different from somewhere where it's 1,000 meters above sea level. Velocity, how fast the water flows, implicates or affects which organisms or this community which might live there. Nutrients, the availability of nutrients. Remember the concept, the river continuum concept? When the river in the middle opens up, the light gets in, it needs a lot of nutrients in order to now provide things like productivity and all that. That could also be driving the change. And finally, the temperature. Some of the organisms might be temperature sensitive, others are moderate, others in there. So out of all what we measured, and so how we interpret these are the variables, the red are the species, in short form because all the name cannot, it's too long name to be in this plot. And lastly, whatever numbers you're seeing in there, those are our sites where we sampled. So if you wanted to say this is a crab, the common crab, which is called Potamonati, will have to be somewhere where there's high elevation, somewhere upstream, almost the source of the river because it eats the leaves, breaks them down. It will be somewhere where the floor is also high because it loves to hide and all that and things like that. So that's what that plot means if you're trying to interpret it. And so we also wanted to see how, you remember the acronym EPT? We said it's an acronym for the mayflies, stoneflies, and caddisflies, which are these taxa, and people use them for biomonitoring the river system. So we wanted to see in the present data which we collected in 3 years ago, how is the distribution of these organisms which are really sensitive to water quality? And this plot tells us almost all of these models are inferring that these communities of these insects, as you go mostly upstream, because elevation, of course, goes up while you go upstream. This is our upstream and that's our downstream if you are having difficulty interpreting this. As you move upstream to where the water is coming from, all the sensitive taxons are reducing in terms of abundance. Does it make sense? Mm-hmm. So which tells you it should be actually inverse true. These communities which are sensitive to water pollution should be more in the upstream. Is it true? Because that's where we postulate not to have a lot of anthropogenic activities. But in this case, this community which is water sensitive, you find in it downstream actually. So what does that mean if you have to use them for biomonitoring them in the Mara River system? So we did a proportion of the same community versus other species which were present and wanted to see what was the trend. And it still confirms the same story. Like as you go mostly upstream, the numbers of these sensitive guys is reducing and inverse true. And so I've already showed you what we study, why it's important. The only point you are there asking yourself, why is it important? Why are these insects important then? Whatever comes there in the bullet, that's how you can use the insects for. And a lot of these are time. The reason why a lot of people use these insects in water for biomonitoring is because the insects are immobile. They move, but not a lot as fish. If anything happens in water, let's say now, if you come and measure even after 5 years, the organisms will not have moved crazily. You can still track them and measure things like ecotoxicology. How are they able to bioaccumulate these different trace elements in their body? They are easy to sample. With your kick net, you can easily just go to the river, do your kick sampler, and voila, you're going to have your samples there. They have rapid life cycle. These organisms in water, they can live between 1 and 4 years, 3 years. And during this period is a long time to really study any change in water, right? So you could use them to study that. They indicate water quality and things like that. The reason why we use them or any other ecological role because they are food source for the fish. When fish are really small, they have to eat these insects before they go to their next stage. So you need those insects in water all the time. They are good with nutrient cycling. The insects themselves helps with nutrient cycling and many more. They have a lot of range of activities or roles, what they play in water. And so this idea, remember I mentioned about how you can use them to monitor river system. So it's not something Joshua is starting now. This thing started way back. In 1964, somewhere in the UK, it dropped all to different countries. And now presently we are somewhere, we are now mostly using, it's more of modified system. The name was actually biological monitoring thing for the UK. So it's the same name, but depending on the region, because you have to use the species of that particular region, the name changes. Because the Kenyan species might differ a little bit with the South African species and vice versa. It's all the same idea, but yeah, the things changes a little bit. So in Africa, we are using this one from South Africa a lot. It's called SAS, which is an acronym for South African Scoring System. And basically what this technique entails in this next page is that every organism has been assigned a sensitivity index. If I'm sensitive to water pollution, I have a really high score. If I'm not sensitive to water pollution, I have really a low score. So the scores range from 1 to 15. Any organism with anything between 1 and 5 is not, it's pollution tolerant, it can live in polluted water, but anything with a score like a 15 really loves to be in clean water. So using this kind of a protocol, you can go to a river, take out your sample. By the help of your taxonomy, you are able to score this sheet, which is called STARS score. And by the end of that session, after you have scored your session, you are able to calculate these two indexes, which is basically the STARS score is the summation of all the sensitivity scores within that site you found, okay? The 15 plus 10 plus 1, that's what gives you the STARS score. Number of taxa, we all know is the number of species. How many different species did you find in that site? If you sum these, you divide it by this number, whatever is an outcome is called average score per taxa. How do we interpret that information then? There's always this plot here with these numbers A, B, C, D, E, F, which is basically almost the same scores like in school. If you get an A, excellent. If you get an E, you might be chased away from the school, you know. So using this, the interpretation is in this next page here that if you sum in A, the interpretation after you sum up that, you divide and you have your SARS score number which you calculated, and you have your average score per taxa which we calculated from here, right? Are you following?
Mm.
You can plot your sensitivities for every site if you have like numerous sites, and that can inform you which site does each site lie in terms of this status. Is it an A? Is it a B? Is it a C? Then what does that mean if it's an A? That means the site is almost natural and modified in that river system. A lot is not happening here in terms of landings. But if you are at somewhere at E, it's seriously modified. You might have they might have developed a dam or there's an industry releasing effluent there. And if you sample the species from there, compare them from another place where it's not disturbed, it's usually different. And this would easily tell you what is happening in the river system. Have you heard of the environmental impact assessment? This is the replica of that in water.
Oh.
This is almost the same stuff in water because in the environment, you measure different parameters. In water system, in South Africa, actually, if you learn this technique, you can be contracted by experts to carry this work to just show them what will happen to this river if you brought an industry, for example. What will happen to species in this river if you dammed this river? What will happen in this river, for instance, if all the trees are cut? So it's the same kind of EIA in water, for sure. And people are getting contracted to just conduct this as their full-time job. People are doing this for consultancy, where the countries are really employing this kind of a technique. In Kenya, we are still at infancy stage. We are not being contracted to do this. But as we showed you the trend of climate change in freshwater system, the focus in 20 years from now probably might be freshwater because everybody needs water to survive. And if we don't have water, people have to learn this technique in order to adapt and also monitor their river system. Like, for instance, here you have Grumeti River around here. You could be the custodian of that data, what is happening now before any modification happens. Like you already have that data even before this modification happens, and you have baseline. Where did we start? Which organisms are in present there so that we can say, oh, well, climate change is happening, bro. It's really happening. This is the proxy to that kind of a measure that you can say, oh, we used to find these sensitive taxas, but they are no longer there in 15 years from now. And so, remember my historical and current sites? We compared, because these sites are, we sampled the same places in different times. We wanted to compare historically, in those 15 years ago, how did those sites score based on the insects which were present there, the species? And as you can see here, is that 15 years ago, all the sites, remember the clusters were really small, which means they almost had the same insects, they almost had the same composition. That was being driven because those sites were almost natural. There wasn't a lot happening in terms of activities in this catchment and things like that. But presently, we have done that along one site, which is inside the forest, has retained its status of natural. The rest of the sites have declined. from an A to a B. Can you follow? From this to that. So we wanted to see what has happened in terms of insects. So we used our data to try to interpret it using this technique. And it's really in support of what we found. Like, and now with this, remember we were unable to define our increase in diversity and decrease. 15 years ago, the diversity was low. But now the diversity has gone up. But from this data, we can see 15 years ago, the sites were natural. So you tell me the increase in diversity means the site is good. Of course it's not. So this data is like supporting what we found in terms of diversity and all that. And so we plotted that, and you can see all the sites in historical time, they scored an A in this plot. And my sites currently are all distributed and even going towards an E. So if you kept this data, if I sampled Mara River for the next 50 years, the trend will be different. It can tell you a lot of story. So what do we do with this information then? I don't want to keep this information alone. That's why I'm here giving this seminar talk. But in Kenya, what am I doing? We are already in talks with a lot of universities in Kenya. We train the students there. We have stations there where we go to the river. We sample these invertebrates for capacity building. We help them identify them. We give them tools to just build their skills in order to follow this path. And they are really enjoying it. We have done this so far in some of the schools or some of universities in Kenya. We have Egerton University, we have done that. At Jomo Kenyatta University, we have done the same training. And at Karatina University, where that's alma mater, where I went to school, I've done this. They came out overwhelmingly. There were so many of them who were willing to learn the technique. And where do I move from there then? I'm planning to upscale that to more like a national reach, if I can reach more people, probably primary schools, probably different organizations, in order for them to understand how this technique works. I'm willing to train people. They don't pay me. I train them free of charge. So if you guys ever want something like this, I'm willing to do it for you guys. So don't worry.
Yeah, so I guess the question is, when are you coming to?
We're going to talk about that. So we're doing this to a lot of people, just the way you monitor species with the community. We're using the community-based organization. We can have the same technique, use the people who live near a river, and help them monitor this river themselves, equip them with the necessary skills and things like that. So apart from just terrestrial animals, next time when you see tourists, tell them this river called Grumeti also has its own species. And for instance, you can print out something. If they come here and you show them what you can find in a river, maybe images of what you can find in river in terms of mayflies or fish species. There'll be, not everybody who comes in Masai Mara is interested, or Serengeti is interested to see wildlife. Others will appreciate more if they saw what was inside the river. Others are ecologists, right? So if you just have a billboard of what you can find in Grumeti River in terms of images, they'll appreciate even more and even bring more people over. And I'll be able to do that because last year I was a recipient of this grant, the WWF Train Fellowship. They have given me some money, which I'll be able to accomplish all the other goals about training people in universities, primary schools, and things like that. And with this, I want to thank people who gave me the permits, Kenyan side, CrossTech, Tanzanian side, Tafili, and all those people, the different funding organizations down there, collaborators, the Dar es Salaam National Museum of Kenya, and everybody who made this research a success. That's our team in the Mara. And with that, time for questions, comments.
It's really nice. Thank you very much, Joshua, for such an amazing presentation. And we definitely learned a lot. I mean, it's quite a new area for most of us.
Yeah.
As you said in your presentation, that most of us have really focused on terrestrial ecosystems, and we can't really have healthy terrestrial ecosystem without freshwater ecosystem.
Yeah.
So thank you very much again for maybe stop sharing.
Yeah. So
Okay.
that you can see the audience.
If they are still there.
Yeah.
Okay, and then stop sharing.
All right. Hey, hi everyone. So if you can do this. Yes, so if you have any question, you can either type on the chat here, or you can just like raise your hand, and then Joshua will be happy to take your questions. Or you can just like unmute yourself and ask a question. Sure, you can ask your question.
I think she didn't hear you. Beryl, you can go ahead and ask your question.
Hello.
We need to mute ourselves. How do we do that?
Okay, can you hear me?
Yes.
Okay, thank you, Joshua, and the team at the University of Florida for the work you're doing at the Mara River. Okay, my name is Beryl. I'm a PhD student at University of Chinese Academy of Sciences in Beijing. So I have two questions. Joshua talked about the issue. From what I've noticed in your research is that maybe you are just considering the aspect of taxonomic diversity when it comes to metrics of monitoring microinvertebrates. So my concern is that from the ecological perspective, you find that when you use taxonomic diversity, as you stated, the Shannon diversity, the PLO evenness, so on and so on, it mostly covers how species changes from the outside perspective, but it doesn't cover the evolutionary of the species. So I was asking, are you considering the issue of functional diversity that tends to be integrated with the taxonomic diversity to get a clear picture of how these species tend to change in terms of traits, something like the traits? And then my second question also, I noticed that you are considering the aspect of the climate change, but there is currently emerging contaminants in aquatic ecosystems. These include the issues of microplastics, pharmaceuticals, and if I'm not wrong, the Mara River is situated in what we term as the dry areas of Kenya. So there's issues of the heat waves, excessive precipitation, and so on and so on. So are you considering emerging contaminants effects on the biodiversity, or you are just considering the abiotic factors that you stated earlier? Thank you.
That's a great question. You need to mute yourself there. Okay, yeah, can you hear me now? Yeah, I think so. So those are really two great questions, and I'll start with the second one, which you asked about if I'm factoring in the trace element or emerging contaminants. Yeah, probably, I don't know if you joined my talk in the beginning, but some of my chapter 2 now will focus on, this is what I presented is just part of my chapter 1 of my dissertation. So maybe next time, George, if you invite me, I can talk more about the trace contaminants, such as mercury in fish, in insects, in water, in sediment, and things like that. And that's something we'll also consider as also an influence to the drivers of these communities as well. But the challenge with some of those contaminants is that they don't stay in water like for all the period you are sampling. You know what I mean? If someone released an effluent today using the pipes, mercury, for instance, into the Mara River, and assume I come to sample after a month, there's been already dilution effect. I won't get the high concentration of that time, you know? And that becomes really a challenge to try to incorporate some of these other different stressors into a catchment. So yeah, we are measuring them. we have that, but the data which we already have from fish water sediment points out that the levels already in the Mara River for trace element, both mercury, lead, cadmium, and so on and so forth, those levels are really so low that if they are so low in some levels, they are not even detectable by the machine, meaning they are really low. They are there, but they are really low. At that point, then I say, probably in order for me not to factor them in is because they were really so low. Even if I brought in them into this model, they probably won't make any change because this 0.00 of ppm, which is parts per billion, which is a small, really, what is, it's like having 1 over a billion. What is that value? Really so small. So some of the times you don't consider, especially in the Mara catchment, I'm not considering trace element as really an impact when it comes to them affecting the composition of these communities. If I will consider functional diversity, that's something we are trying to explore. And I think by the end of the day, we will have matrices to try to also understand more on the functionality or the functional diversity of the insect as well, apart from using Shannon Weiner, of which at this point it is still factors in the species richness and also how many of each different type of insects there we have in water. If I answered your question, very.
The first question, I think I will also add a little bit. I'm not very like expert, but I think what Joshua has done is to have like fundamentals of what, like the baseline.
Yeah.
And I think the following research question will be to look at the functional diversity of where you start looking at adaptation of species. So for example, if you have those like indicator species that he mentioned, then you look at evolutionary adaptations that has been going on at molecular level. But I think what his work has really demonstrated is mostly like a baseline in which we will have like future reference.
Yeah. In addition, also, we are considering, this is just a part of what I do, but we measure different aspects such as isotopes, how much of nitrogen isotopes and carbon is there in this water? Where does it come from? Is there niche petitioning between these species? What is really happening? Like we have a lot of data on other different aspects, which probably haven't talked in this for today.
But thank you for very nice questions.
Need to mute ourselves now.
Yes, Juma, you can ask your question.
Yes. Thank you very much, Joshua, for
Juma,
such a nice work.
Can you
yes,
hear us?
Can you hear me?
You are muted, I think.
No, I'm unmuted.
Oh, here.
Can you hear me now?
Yes.
Yeah, thank you very much, Joshua, for a nice presentation. I must admit I'm not competent in this field. So I have 2 questions. Number one, maybe I skipped understanding this. Do we have like a clear gradient on what is happening? Like maybe in the upstream, it's better getting towards the downstream, like the pollution level. But number 2, I'm also aware of the field work that was done in the Tanzanian side of the Mara system. Even though you stated that this is still work in progress, can you give us a clue on what is happening in comparison to the Tanzanian side? Sorry, in comparison to Kenya side. Thank you.
Can you hear us? Can you hear us now? Yeah,
Yes,
I think
can
so,
hear.
right?
Yes, please. Yes.
So I think Juma, I probably have met you somewhere in Musoma, if I'm not wrong. Okay. So the drivers of if you read the book chapter which I've recommended, which entails what is happening in the catchment, you will have this conceptual framework of really what are the stressors in the Mau forest. I've used Mau forest as one of the case study in this particular example. So I recommend, or you can write me probably inbox or whatever. I'll send you the old paper and probably read it. But there we have all the drivers. Coming to your second question about what does the data in Tanzania compare to that in Kenya, for this particular study for diversity, because I wanted years, as you have seen, there was something done in 2000 and 2008, right? 2008 and 9. I've sampled in 2021, 2022, same sites in Kenyan side. In this particular study, I've excluded Tanzanian site because I do not have any sites which were sampled during that time. My data for reproducibility has to be the same. So for this particular paper, I'm only focusing on sites in Kenya. But when it will come to issues of trace contamination, how much of mercury we have in the Mara River, then I have the longitudinal gradient of study from all the way from the Mount Forest to Musoma Bay, which is where the river flows. And that is my chapter 2, which I'm sure at some point, Juma, you might be interested to see what I found, but I'll be signing up for this kind of talks if they ever have any other chance for me to talk about the trace contamination. So for this particular study, no, we never included Tanzania because we wanted data for 2 complete years, and we have never sampled in Tanzania.
Thank you. Kaiser?
Yeah, yes, please.
Yes, you can go ahead and ask.
Yeah, so again, thank you so much, Josh, for such a wonderful presentation. It was informative. I missed a big part of it, but my question is, we know how important the Mara River is to the survival of the Serengeti ecosystem in general. And somewhere in your results, we see an increase of microinvertebrates within the Mara River ecosystem itself.
Yeah.
So as conservationists, should we be worried of the increase, or are the increase very, like they have neutral effect as far as, you know, the terrestrial mammals are concerned?
Thank you so much for your question, Kaiser. You actually reminded me of a very important point I didn't point out. I won't have time to go back to my map, but as you saw my study area, the site number 7, 8, 9, because we have 9 sites in Kenyan side, the most downstream sites are sites within the Conservancy or Masai Mara Park or Reserve in Kenya. That area is where the wildebeest crossing occurs or happens every single year. You have brought a very important point about you remember what I had issues explaining my increase in diversity in some of the sites, especially the most 2 sites which were site. I almost the last site. The last site is at the point between the border between Kenya and Tanzania. So the site number 7, 8, 9 accounts in for the all influence of what I'm calling wildlife subsidy input. Within that section is where the wildebeest are crossing. They will die within this river system. The nutrients are high. Insects will want to colonize such area because it has high nutrients, because they are looking for algae and all things like that. Fish will want to come there because catfish will love to go and munch or eat that kind of free meat there. So that area brings in a different biodiversity as you can ever compare in other river system. An example of Mara River is a typical different river. Any other river like Grumeti River will have a clear probably patterns. But the Mara is unique in this way because where we have the influence of the wildlife, more hippos, where they defecate in the river system, their dung decomposes, brings in the food web, it's part of the chain, we're having more abundance and diversity of these insects. That is an important point I forgot to mention. So Kaiser, thanks for bringing that up. And you shouldn't be worried about that. That has been brought up by natural occurrences. The wildebeest have to cross the river, they'll anyhow die because they'll be attacked by crocodile. Others jump from cliffs, the cliffs which are not really good, and they're gonna break their legs. They are unable to walk. They just die in the river system. So it's something you can't really control. So it's more like a natural occurrence, which there are those kind of animal legacy kind of stuff in natural ecosystem. Yeah.
So from those who are in person, if there's anyone with question to
Yeah, sure. You either probably understood everything or I didn't do that great. Either of the two. So prove me wrong.
So for me, mine is little general. I'm just like thinking of like a collaborative project with Bumeti,
Yeah.
especially because we have two rivers here. We have Rubana River, which is just like a few meters from here.
Yeah.
And then we have Bumeti River, and I don't know, from your literature review, probably you have seen nothing has been done to kind of like
Almost. So the weird thing for the rivers, remember, everybody comes from a village unless you are born in the cities. Well,
yeah.
It will be next to negligible if your river has been studied from where you come from, because we have less scientists who are interested in freshwater. Everybody wants to rush to the terrestrial ecosystem. I never gave a talk about how I got myself into freshwater. That's a talk for a different day. But I was a terrestrial guy. I love to study birds. I loved to study, I still do. I can write a proposal on birds, vultures, or elephant species. But I've now been drawn more into this ecosystem, which is less studied, more critically threatened by multiple threats, less taxonomists, less everybody, because for some of us, I wouldn't say we are lazy, but we don't want to work under microscope. How will your day be if you came here at 8 o'clock and what I find in your desk, you have like a microscope that excites you to just have a microscope there and play around identifying stuff. So not everybody who loves that, but if you are that kind of a person, then I will point you to this direction, which has less people. It has made me to be who I am. I've traveled the world because of what I'm doing. So it's important. And it's equally important as terrestrial ecosystem as well. Yeah.
Yeah.
I'm willing to do all what you have said.
Yeah,
we can have a picture.
Yeah, sure.
Yeah.
Thank you so much. This was really interesting, and I learned a lot about
The freshwater
freshwater ecosystem.
Yeah.
And so I saw you some of the the factors that were influencing this diversity.
Right.
And I was thinking, how can you tear them apart, or which one is the most important as compared to what you have done?
Yeah. That's a really important point. When you use this kind of analysis, there's something which it's called fast forward selection, whereby you can introduce your elevation, you can introduce multiple other variables, and try to measure how does each variable affect what you are studying. The problem with what I do in freshwater system, a site does not exist in a vacuum. If I'm study from here, this is my site 1, my site 2. A river is a continuum thing flowing, right? So apart from site 1, any other site from site 1 will be affected by what is happening in upstream. You get my point?
Yeah.
So it's difficult to tear down and say the influence in my last site is because of land use only. Because it rained somewhere upstream, it brought in nutrients from farms, pesticides, everything. So in freshwater system, it's next to impossible to try to decouple the effect of one from the other. That's why it is a merge of these factors. But as you've seen, I've really taken time in terms of land use. We have factored in a lot of factors. We just want to see in every point, what is the drainage basin of that catchment? And with that, we are able to say, oh, land use really has a greater impact upstream than downstream. While the wildlife subsidy input into the river, Amara River, it only happens in a small section of the river, not the whole system. So sometimes, and that's what drove me to say the increase in diversity in downstream, which it should not be, was brought about by this other variable, which is unlike in any other river, which is like wildlife. So in that case, I can say land use is a constant thing in my system, but how about this other variable called wildlife subsidy does not exist in every site, it's only available in these sites. And then I could say, oh, well, this then might be driving my changes in this particular area.
Thank you.
Pollution is happening in all the catchment. We have launches and hotels along this catchment because it's touristic. I don't know if this is the case of Serengeti, but you find that these launches, where they keep their tourists, somehow their effluent from these different places end up in Mara River.
Well, there's a question from Raleigh. Can
Yeah.
just like answer that there.
Yeah, that's a good point. I think really, Beryl, she's a truly ecology. She is understanding the whole. I'm sure she's Kenyan. She understands what's happening. She understands what's happening in the catchment, and she's putting me on the spot for sure. And it's a good point because yeah,
She's preparing you for your thesis.
yeah, it's a good question. This is a good question. Yeah. So issues with grazing, pressure, and all that are all coupled in this one big stressor I called anthropogenic pressure. If you see this catchment where I'm studying, we have large numbers of cattle from Maasai community, which they are grazing, they make the land more susceptible to soil erosion and things like that. Or even when they go for watering, they are disturbing all the environment in the water all across the catchment. And so, just like I said, we are factoring it as a part of anthropogenic pollution by how do we do that? In all our sites, we could count and say the number of active activities. We go there when you are sampling and we watch around. Are there people coming to draw water from the Mara River? An estimate of how many cows during that time we sampled are coming for watering events and things like that. So that somehow also has been accounted in our those more in form of anthropogenic disturbance.
Yeah. Thank you very much. No. Yes, we
So
still
to
have.
speak, thank you for great presentation. It's
Yes,
quite informative for most of us.
Yeah.
Like it's been said already. So mine is about first of all whether the serious I have 2 points.
Yeah.
I wanted to just ask whether the serious flooding events.
Yes.
Where like the riverine,
Yeah.
the river banks are seriously discovered, and everything like really shaken.
Yeah.
In that river, the water flows from the forest downstream. Whether that will influence what you find depending on when those sampling activities took place.
Yeah.
And the other thing, referring to your point about light. Like if the light penetrates and it influences what happens in the water. Whether you're looking at how intact the riverine vegetation is then and now, or when if one does that, like if we that's something one needs to take into consideration.
Yes. So I'll start with your point one, which is I'd say yes. Issues of any events like big events such as flooding or whatnot affects what you might find. Why am I saying that? You saw one of our projects in the Mara is tracking the fish. So we put these trackers in this fish back in 2023. Then came this big rain, which almost displaced everybody in Masai Mara. And everything was locked down. And since then, our trackers outside these antennas are unable to detect our fish. So two assumption hypothesis, they might have been swept all the way downstream, 1, 2, they might have died. Somewhere downstream and they are unable to come back and to be detected by these tracers, by these antennas. Same thing happened with organisms. These insects, when it rains, not that they don't go to the gym now that they can bend on stop like I'm not gonna go when this flow. They'll be swept down. And they have the chance now to come back slowly colonizing these different areas. So that will affect. And probably during our sampling time, there were more events of flooding than during 2008, 2009. So that's another variable, but again, it will be difficult to account for it, like how much magnitude does that really impact what you are finding, right? The other question was about the riverine ecosystem. Yes. So if we ever do this for macroinvertebrates, there's another technique to score your habitat. So before you sample, you're gonna stand outside and score your habitat. How much is the forest cover? 80%. How much is whatever? So you usually have that data being supported by these habitat, whatever is happening within that particular site. And we have a technique called cleanan, which is more habitat suitability. You assess the suitability of your site first before you even sample, which we did account for, yeah.
Yeah, I know, like, this is very interesting topic, but we will have to end here because of our time, but we will have we will send Joshua's email to everyone for those who will be interested to reach out to for him to share his articles as he mentioned during his presentation. Yeah. But for those who are on site, we have like another few minutes to chat with Josh. But thank you very much again, everyone, for your attendance, but also a big applause to Joshua for your presentation. Right. Thank you very much. See you next month for another seminar talk.
Thank you.
Thank you, George.
You're welcome.
Bye-bye.